Okay, awesome. Yeah, so I am going to jump right in here then to make sure that I don't take too much time. Thanks Patricia for the introduction and the organizers for the opportunity to talk here today and speak in this session. I'm excited to tell you about some of the work that I'm doing with Paul King and his group and understanding kind of our broader interest in the physical biochemistry and photosynthesis group is understanding fundamental principles of protein control for multi-electron redox catalysis. That's like our mission statement I have over there. But we're kind of really focusing in on using the iron-iron hydrogenases, which we all love, that catalyze the reversible activation of hydrogen as kind of a model framework within which to study different elements of enzymatic control over reactivity. So the iron hydrogenases do this activation, utilizing the same Kelly cofactor, the H cluster, and have similar activities, but there are other elements of the protein, of the enzymes that are different and have a lot of diversity that's shown them. So I'm just real quick for our background. I really just want to kind of illustrate or show what those are. So we know that the proton coupled electron transfer chemistry that happens, that happens at the H cluster active site. So there's control that needs to happen there of the incoming and outgoing electron and proton fluxes and kind of making all of that work together. So that's one aspect. Then we have the network of residues that allow the proton transfer in and out of the active site. So that's another aspect where variation is seen in those residues, and that can be investigated in terms of how it contributes to reactivity. And then the electron transfer that happens in and out of the active site as well. So these electron relays that function to direct the electron flow in and out. And what I have shown here is just a minimal model of iron-iron hydrogenase. This is like example from the Clemens Reinhardt. So it's very minimalistic in that it doesn't have any really additional subunits or electron relays. It's just got direct binding to this ferredoxin partner. But there is a lot of diversity in those electron transfer relays. So that's really kind of where I'm going to be focusing. And we're kind of homing in on looking at those relays and how that connects to reactivity control. So first, I just want to also orient ourselves with the different components that matter in biological electron transfer. So I know that this equation is very familiar to everybody, but I just wanted to put it up here and kind of pull out the components to make it a little bit more simple to look at. We have things like the thermodynamic landscape, the reionization energy, and then this HAB term, the electronic coupling, that all are important in the control or in the rate of electron transfer. And this has been looked at a lot in biological systems. So thinking about more broadly, when we think about electron transfer, we often consider these in terms of different parameters that can be tuned, and we can observe how they contribute to different observable rates of transfer. And this has also been done in redox, or this has been applied in a lot of redox enzymes. And specifically, I want to draw attention to the studying of examples of electronic coupling in these enzymes. So this has been done in complex 1 with mapping of the iron sulfur clusters by EPR, spin-spin interactions between the nickel site and the iron sulfur centers in nickel iron hydrogenase. And in the iron-iron hydrogenases also, there's been work done by Patricia on the DD organism and the CA1 and CP1 iron-iron hydrogenases that have 4 of those accessory kind of iron sulfur clusters. So as an example of a lot of a more complex, electron relay there. And there we have also site differentiated clusters. So all of these things are examples of studying these electron relays in these systems and also looking at how the electronic coupling of them can influence or impact reactivity. And we know that coupling is directly proportional to electron transfer. So it's something that we want to examine a bit more closely now in the CP Clostridium pasturinium hydrogenases. And why this is is because the CP system, we believe is a unique model system that we can use within the iron-iron hydrogenase family to examine aspects of reactivity control. So for the electron relays, and then also in those other elements of the protein, there is diversity in the electronic structure of the H cluster between these three hydrogenases. They have distinct activity biases, which I have shown down below, and the complexity of the electron relays. So CP1, like I mentioned, has the 4 iron sulfur clusters there in the green domain that represents the kind of electron transfer region. And CP2 and CP3 both have 2. However, CP2 in particular, the iron-iron hydrogenase 2 in this organism, we're interested in because it has this high directionality to its reactivity as well. So it really favors the H2 oxidation reaction versus the reverse reaction backwards. And so we really want to focus in on looking at the CP2 hydrogenase as a model for examining what role the electron relays might play in electron transfer and perhaps in the directionality that we see in CP2. So there's been work done on CP2 already. There's a really great body of work that was begun in the 70s and 80s and kind of into the 90s a bit, spearheaded by Mike Adams. And they had, so I'm kind of going to describe what that is in addition, while also going through our current results to kind of remind everybody of, I guess, where we're at in our current knowledge of the system. So these 2 iron sulfur clusters that I'm going to have kept up in the top right, just so that we can keep oriented. There's a proximal one nearest the H cluster, and then a distal one on the outside. And Adams and coworkers, when they initially started looking at the system, they got the EPR, and they were two different distinct kind of regimes. They noticed temperature really made a difference in the EPR signal that they observed. So I also have then following up on this work. So we see that when we have just the CP2 hydrogenase reduced with hydrogen and collecting temperature dependent data, and I have shown selected traces here. The higher temperature, there is this kind of very characteristic 4 iron 4 sulfur cluster signature. I have that the middle turning point there indicated at 1.93 that Adams had pulled out in his higher temperature data. So this is kind of, again, like I said, characteristic of an iron sulfur cluster signal. Made sense that because we have 2 iron sulfur cluster accessory clusters. But at lower temperatures, what they noticed is that there was a lot of additional spectral complexity that grew in at the lower temperature. And I'm going to step through what those are. So we see the growing in of this feature at G equals around 2-ish. So 2.01 I have indicated here to match with what was indicated previously. So that shows up at the low temperature. There is this excessive broadening on the high field side. The point that I have indicated right there is about 1.79, I believe, for a G value. But there's this really, really excessive broadening, which has also been seen in iron sulfur cluster systems. And then this feature at, they had indicated around 1.9, and I have right there that grows in at low temperature as well. So all of these additional signals at the lower temperature were collectively ascribed to the second iron sulfur cluster that is known to be in CP2. So from this data, it wasn't just EPR, there was a whole suite of kind of biophysical techniques that went into also kind of ascertaining what they could about these signals, looking at the potentials of them as well. With the higher temperature signal, they potentiometric titration of that kind of middle region there between the second and third turning points was able to pull out a potential of -180. So I'm going to move here now to here I've shown my data now separated to match with theirs. So the higher potential cluster that was from a potentiometric titration, monitoring that higher temperature signal. And then the potential of the other cluster has been bracketed at lower than minus 300 millivolts. And that kind of came about from a combination of different pieces of data. Initially, it was MCD, parallel MCD and Mossbauer that showed that the signals in the EPR could be linked back to two distinct S equals 1/2 4-ion 4-sulfur cluster signals. And in a sample that was poised in an intermediate redox state corresponding to about minus 300, this EPR signal that shows up at low temperature, this complex signal that I went through on the previous slide, it was absent. It was absent in the sample poised at minus 300 millivolts, and the Mossbauer of that sample also showed absence of about half of the paramagnetic contribution compared to a fully reduced sample. So those two pieces of evidence together led to this lower temperature signal that I have shown here being ascribed to the F prime cluster, as I'll call this second lower potential cluster. And like I said, it's bracketed at some potential lower than minus 300. So this is kind of where we're at, right? Or this is the current proposed model here. And then I want to just take a slide to show exactly how this fits into a landscape model of then electron transfer in CP2. So this is the pathway that, one of the pathways that is possible here proposed has the high potential cluster on the outside and the outer location, sorry, as the distal cluster. And then we have the lower potential, this F prime cluster that is exemplified by this very complex lower temperature EPR signal is in between the H cluster and the distal. And this model would favor, has a favorable thermodynamic landscape for hydrogen oxidation, where the electrons, as they are getting shuttled out, thermodynamically move towards that distal cluster, and really the barrier then just becomes a transfer to the ferredoxin partner. Another model could also be considered then, where the roles are reversed, and the higher potential cluster now sits in the middle location as the proximal cluster to the H cluster, and the lower potential cluster sits on the in the outer location as the distal cluster, where it is closer matched to the ferredoxin potential. So either one of these models could be correct. The initial model put forth on the left, or this one on the right. I'm not necessarily showing anything that's going to say one or the other, but I think that the remainder of the talk, I want to show kind of some evidence that we have that makes the model on the right equally plausible. So how am I doing on time? Okay. So these models kind of really differ where the thermodynamic barrier is. Which model makes more sense? To do this, I want to go back to the data that I showed and look more closely at those, at that complex lower temperature signal. So before we really could even begin to look at these additional features and try and pull out what information we could about them, we had to address the fact that we really don't have a defined signal for the second F cluster that is that we can simulate. So everything's overlapping. It's really complicated. So I turned to using Q-band, so multi-frequency EPR. So I did Q-band EPR, pulse Q-band at different temperatures to kind of start to recreate that temperature dependence. And here I have shown the 3 different temperatures of the same type of sample reduced with hydrogen. And I have the G values on here, but I just wanted to show them all on top of each other because it's easier to see the differences in temperature before I show the simulation. But you can see that there are changes with temperature that are similar to the changes that you see at the CW. And in fact, when I do a global simulation of both frequencies, I'm able to resolve a second rhombic G tensor that I can assign to the F prime cluster. So before, this additional F prime cluster was just kind of assigned to a suite of signals, all of these different signals that showed up at lower temperature, but it wasn't really clear what exactly the G values were. So here, by simulating both sets of data, I can pull out the contribution from the F prime and the F cluster and confirmed that rhombic EPR signal that was previously identified for the F cluster with G values that are pretty close to what was originally stated. So now, having kind of a firmer grasp on what the F cluster signals are, we can go back to that CW data and we can say, okay, those are the middle, where we see a lot of changing in the middle here. Those are where the two different F cluster middle G values show up. But what about these other two components? What about this signal at 2.01 and this high field broadening? So these are still unassigned. We can do an initial way to kind of look at pulling out information about whether signals go with each other or what they might be from is looking at the temperature dependent relaxation profile. So having the temperature dependent data like this, we can create relaxation profiles for these various species. First, if we look at that done for just the F cluster signals, which I have here, we can see that they are distinct from one another. And I guess I should also explain real quick, what you're seeing here is a plot of the signal intensity that I take at each G value. It's normalized for the dependence on the Curie law, and then I plot it versus temperature. So if it's a flat line, it indicates that the intensity is changing as you would expect with temperature, and there's no saturation behavior or like relaxation or temperature broadening, and changes that are not a straight line indicate something else, like what I just described. So monitoring both of those signals, we see that they do have distinct temperature dependent relaxation profiles, which we would expect for them being from two different iron sulfur clusters. But really then looking at these other signals is kind of where it became interesting, in that looking at the unassigned, I guess you could say, signals, in the bottom graph, I have them all shown together. They all have similar profiles and a similar dependence on temperature. And the top, I'm just showing two of the signals compared to the F cluster signals. Their profiles appear distinct from those of the two iron sulfur cluster assigned signals. So the second iron sulfur cluster, that F prime signal that shows up at the low temperatures, it has a similar behavior as these unassigned signals at higher temperatures. But at the low temperatures, we see a deviation that happens where, let's see, can I put probably should have my laser pointer. Pointer options. Okay, well, anyway, sorry, I couldn't find the laser pointer quickly. I'm not used to Zoom anymore. We have to use Teams here at NREL. But anyway, the top graph, what I'm saying is that at the very first few points at those low temperatures, you see a distinct behavior where the signal at 1.91 is pretty flat, and the behavior for those signals in teal, these unassigned signals, instead is on a curve, indicating that there is saturation happening at the very, very low temperatures for those signals, unlike with the F prime cluster signal. So there's a lot more that you can do with temperature dependent data, and a much more, more quantitative results that you can gain. And that does form the basis of work that we're interested in, as you'll see on my future directions slide. But for right now, these offered a nice qualitative picture of helping our assignment of these different signals and the F versus F prime G values, and then these unassigned signals that seem to have distinct relaxation profiles from the F cluster signals. So in the previous work by Adams, and hearkening also back to the work that I described in the nickel ion systems and et cetera, these signals were hypothesized to be manifestations of electronic coupling between the F clusters, which is supported by the distinct relaxation behavior that we see from the F cluster signals. And going back to our pathway that I showed in the beginning, I want to kind of bring this together. So we've resolved the signals from the two different F clusters and have evidence of this additional spin coupling now. We've been able to distinguish the F cluster signals from these unassigned ones that are attributable to spin coupling from electronic coupling between the two clusters. What does that mean in terms of these models? Well, like I said in the beginning, I'm not really showing anything that is refuting one of these models or the other, but what I am hoping to do is show that this evidence for this kind of strong electronic coupling that we see in CP2 provides a way to think about how the electron transfer relays might be aiding or might be functioning in the role of in the directionality of the reactivity that we see in CP2. So here I just want to show this figure from a paper in Nature in 1992, Page, Moser, and Dutton, where they were looking at electron transfer rates in these different systems. And here they had this initial image from a nickel iron hydrogenase into desulfovibrio-gigas. I didn't want to get that wrong. That's why I put it on the slide. But they show this really cool landscape, right? Where they have this, if you look at our pathway 2 on the right, similar setup where they have the nickel iron center and then this really kind of thermodynamic well where they have this high potential cluster that is in between the active site and the external redox partners. But in this system, they calculated that the rates of electron transfer that they calculated over tens of angstroms were in a range that was like an order of magnitude more even than the experimentally determined electron transfer and catalytic rate. So this setup alone and the ability of the electron tunneling in the protein, like the enzyme matrix, was able to account for the electron transfer rate seen in the actual system. So we can try and map now, very crudely, I have just shown here what this pathway 2 could look like on this scheme where we have our H cluster, we have this high potential cluster now in between the H cluster and the distal cluster. And it follows kind of a similar scheme that you see in this nickel iron hydrogenase, which also has the nickel iron hydrogenases have this high directionality towards the H2 oxidation that we also see in CP2. So a setup like this could provide a barrier to the reverse reaction, providing some context for that directionality that we see. And it could be linked to the electronic coupling that we have manifestations of in our EPR data. So future work this is really just the tip of the iceberg that we're touching here with thinking about a different way a different kind of framework to think about the F clusters in CP2 and really how they might be contributing to reactivity in general. So we want to use advanced pulse EPR techniques to further investigate these aspects. I have shown here on the right, this HAB term that I discussed that's in electron transfer can be decomposed to the components for magnetic dipole-dipole coupling, as well as electron-electron exchange coupling. Pulse EPR is great for looking at those types of components. So we want to start to kind of build an approach of doing that within the CP2 system, and couple that with some other things like mutagenesis, perhaps, to try and get some additional, more direct evidence for the placement of those clusters within CP2. We're working without a crystal structure of the enzyme or anything. So we're trying to do what we can with the different structural techniques that we have to map as well as we can where things are located, and use all of this together to kind of expand on the model that we have of enzymatic reactivity in iron-iron hydrogenases in general, and try and draw connections where we can to other enzymes as well. So that is the last that I have there. Acknowledgements here are funding at U.S. Department of Energy, everybody in the biochemistry and photosynthesis group, our collaborators. I got this slide from Paul this morning. Thank you very much. So some of these collaborators might be on the nitrogenase project. Sorry about that. And that is it. Thank you, everybody. And